Welcome to the Wright Science Daily Revision Boost. In today's Biology Blast, we're going to have a look at the different cycles. So the first cycle is the nitrogen cycle, shown by the diagram on the left there. And the key thing we need to remember are that we've got four groups of microbes. So the decomposers, which are bacteria and fungi, turn proteins into ammonia. The nitrifying bacteria turn ammonia into nitrates. Nitrogen-fixing bacteria turn nitrogen into nitrates. And the denitrifying bacteria turn nitrates into nitrogen. Our second cycle is the water cycle. So if we start off in the ocean, then water will evaporate as the sun heats that surface water. So it becomes water vapour and rises up in the air. As it rises, then it's going to cool, and therefore it condenses back into liquid water droplets to produce our clouds. And then as those clouds cool, eventually the water droplets get heavier and will fall as rain in the process of precipitation. That water then runs down the surface. Some of it will percolate through the little gaps in soil and rocks to end up in our groundwater. Other water will be taken up by our plants, and then that will be lost from plants in the process of transpiration. Our third and final cycle is the carbon cycle. So this is just the process by which carbon is cycled through the atmosphere, the earth, plants and animals. So the diagram in the bottom left summarises it for you. We've got three processes that will release carbon dioxide into the atmosphere: respiration, carried out by all living things, decomposition and combustion. And there is only one process that removes carbon dioxide from the atmosphere, and that is photosynthesis, that happens in all our green plants, algae and the phytoplankton. We do find that the carbon dioxide levels will vary throughout the day because photosynthesis only happens in the light. So that means carbon dioxide is only removed during the day, whereas respiration happens in all living cells all the time. So we release carbon dioxide at a consistent rate. And don't forget that human activities are also to blame for increasing the amounts of carbon dioxide in the atmosphere. So that would be things like the increased number of factories, more cars on the roads, greater amounts of deforestation. All of those contribute to increasing carbon dioxide in the atmosphere. Today's chemistry check is looking at how we can test for different chemicals. So the first section, we need to know how to detect different gases. So for carbon dioxide, we're using lime water, which goes from colourless to cloudy or milky. So lime water is a solution of calcium hydroxide, and what happens is the calcium hydroxide reacts with carbon dioxide to produce water and calcium carbonate, which is a white precipitate, and that's what we see as the reason that it goes cloudy. Second test is for chlorine. So chlorine dissolves in water to form an acidic substance, and what it does is it bleaches substances, changing them from coloured substances to colourless. So to test for chlorine, we use a drop of tap water to dampen blue litmus paper, and then when we hold that paper near a container, if it goes red then white, we've got chlorine. Last two tests, we've got hydrogen. We get a lit splint into the tube and then it makes a squeaky pop sound. Or the test for oxygen is a glowing splint will relight when it's placed in a tube with oxygen. Just one of those safety precautions that we could be asked about: never inhale a gas directly from the container. You hold it a few centimetres away and waft the smell towards you to take a cautious sniff. The next test we're going to look at is the flame test. So when metal ions burn, then they're vaporized. So they absorb energy and the electrons that make up its structure jump to higher electron shells. And then as those electrons drop back to their normal electron shell, we transfer light energy to the surroundings. Based on the colour of light that's produced, we can then identify what metal ion is present. So in order to carry out a flame test in the lab, you get a nichrome wire loop, you dip it in concentrated hydrochloric acid, and then you hold it in the blue Bunsen flame until it doesn't change colour anymore. Once we've done that, we dip it into our test powder or our test solution, hold it in the roaring flame and note what colour it is. There are 5 colours we need to remember here. Lithium is red, sodium yellow, potassium lilac, calcium an orange red and copper a green blue. Just remember that one of the possible sources of error in this one is contamination. Third test is our hydroxide precipitate test. So group 1 hydroxides are soluble in water, whereas most others are insoluble in water. So we use sodium hydroxide because it is soluble in water. Now when sodium hydroxide is added to a solution containing metal ions, we form a precipitate, and the colour of that precipitate tells us what metal ion is present. So the colours we need to know: iron 2 is green, iron 3 orange brown, copper 2 is blue, calcium and zinc are both white. The way we can tell the difference between calcium and zinc is zinc hydroxide will dissolve and form a colourless solution if we add an excess of sodium hydroxide, whereas calcium hydroxide won't dissolve. I've given you just the equations at the bottom there just to model these reactions, just in case you want to go over those. 2 words we need to know the meaning of first of all. Anion is a negative ion because it's attracted to the anode, and a cation is the positive ion attracted to the cathode. So when we're detecting anions, we're detecting negative ions. First one, sulfate ions. What we do here is we add a few drops of dilute hydrochloric acid first of all, and then add a few drops of barium chloride. If a white precipitate forms, we have sulfate ions present, and I've given you the equation at the bottom there just to show you what's happening. To test for carbonate ions, we just add a few drops of hydrochloric acid, and if bubbles of gas are made, carbonate ions are present because those bubbles of gas are carbon dioxide. The last test we need to know is the test for halide ions. So to do this, we add a few drops of dilute nitric acid to remove other ions present that could create a confusing precipitate colour. We then add a few drops of silver nitrate solution, and depending on what colour precipitate we make, will tell us what halide ion is present. So if we make a white precipitate, it's a chloride, cream is bromide, and yellow is iodide. In today's physics prep, we're going to have a look at sound. So when a wave travels from one medium to another, its velocity can change, and so can its direction. So this is refraction. So wherever we talk about refraction, it's a change in velocity. If our wave is moving from a less dense medium to a more dense medium, then the wave is going to slow down and bend towards the normal. So this is going to decrease the wavelength. The frequency, however, will stay the same. If our sound wave hits the boundary at 0 degrees to the normal, it will change speed, but it won't change direction. As the wave hits a boundary, one of 3 things can happen. First, it could be reflected and gives us an echo. Secondly, it's transmitted, which could have refraction involved. And thirdly, it could be absorbed. When we're talking about ultrasound, this is sound with a frequency of greater than 20,000 Hertz. And humans can't hear this, but other animals can. Now, ultrasound can actually be quite useful to us because it's got a short wavelength, which means we can focus it into a beam. When we're thinking about how we can use ultrasound, the most common one is that we can scan unborn babies, but we could also use it to find kidney stones and monitor blood flow. So the way that this actually works then is that we use a transmitter that beams the ultrasound wave into the patient. As those waves hit surfaces inside, they're reflected back from those different boundaries, and then as they're detected by the machine, it calculates the distance using time and velocity to create the images on the screen of the baby, for example. We can also use ultrasound for echo sounding and sonar, and that's going to allow us to calculate the depth of water. If we actually shout in an empty room, initially you hear an echo, but eventually that sound fades away. So what we find is initially the sound is reflected, but it will eventually be absorbed, making the particles in the wall vibrate and makes the wall a teeny bit hotter. If we think about the human ear then, the ear detects, amplifies and converts sound to an electrical signal. So the first part, the pinna and the auditory canal, gather the sound wave and direct it to the eardrum, which then vibrates. As the eardrum vibrates, the ossicles, which are the three tiny bones, the hammer, the anvil and the stirrup, will vibrate, and they also amplify the vibration to pass it to the inner ear through the oval window. Once it's in there, we've got the cochlea, which is filled with a fluid, and that transmits the vibrations from the oval window to small hairs on the cochlear wall. Those hairs are attached to sound detecting cells that release chemical substances to trigger a nerve impulse down the auditory nerve to your brain. Your brain then processes the signal and you hear the sound. A couple of key terms we should know when considering sound in the ear. We've got natural frequency, which is the frequency at which an object oscillates if it's displaced. And we've got resonance, which is the large amplitude oscillation that happens when you make something oscillate at its natural frequency. So the hairs inside your cochlea have different lengths and therefore resonate at different frequencies. So the range of hair lengths determines the range of sounds that you can actually hear. One of the downsides of getting older is those shorter hairs are lost, and therefore you're going to struggle to hear those higher frequencies. Hence why places can use that mosquito sound to irritate teenagers, but the adults are happily wandering around without hearing anything. Now we've had a quick recap, then head on over to socrative.com and test your knowledge on the topics from today. The room name you need to enter is Wright 2662. Good luck! As always, if there's something you wish to look at in more detail, then today's topics are for biology in the B4 playlist, for chemistry the C4 playlist, and for physics it's only for the GCSE physics guys today, so that means it's the P5 playlist.